Ukrainian Neolithic female, identification I1732. I cannot say those names of the village, but it's in the Ukraine. I match 0.19%, genetically closer to the sample than 85.48% of other genome link users. This sample from a Neolithic female individual unearthed at the Vovnigi Neolithic Cemetery archaeological site, positioned near Vovnigi village in the region of Ukraine, on the northern bank of the Dnepropetrovsk River. The cemetery is a collective burial place where the remains were found in long trenches. Her skeleton remains were radiocarbon dated to be from 5364 to 5213 Cal BCE. DNA extraction was performed from her petrous bone sample. Her mitochondria DNA haplogroup was identified as UB4. The prehistoric human population started contributing to the gene pool of present-day Europe in the Mesolithic. In the Mesolithic period, there was three main groups of hunter-gatherer populations. The so-called Western hunter-gatherers emerged from a West Asian or East European glacial refugium after the last glacial maximum, around 14,000 years BP. They migrated into West Central and East Europe via the Mediterranean coastline and the Danube River. The other group was the Eastern hunter-gatherers, who mainly descended from the Upper Paleolithic ancient North Eurasians, who in turn received a limited amount of genetic influx from the Western hunter-gatherers. In the south, from the Caucasian hunter-gatherers, the Neolithic transition / revolution and the adoption of farm and life substance started in three Near Eastern centers: the Levant, Iranian Zagros Mountains, and Anatolia. The Neolithization and spread of farming culture into Western Central Europe, you can tell I've got no go with big words, happened through endemic diffusion with limited admixture between the Neolithic newcomers and the local Western hunter-gatherers. The scenario in East Europe and North Europe was different. The adoption of farming technology happened without the genetic influx of the Anatolian farmers. It took the form of a cultural transition. The I1732 individual is a good example of an Eastern European Neolithic population without affinity to the Anatolian farmers. On the principal component analysis, PCA, the Ukrainian Neolithic samples are closest to the Ukrainian Mesolithic samples. Baltic Neolithic samples occupying an intermediate position between the Romanian hunter-gatherers, Iron Gate hunter-gatherers, and the Caucasian hunter-gatherers. They also cluster near the Ukrainian Neolithic individuals. Why these big words? Regarding their genetic composition, the Ukrainian Neolithic individuals show 43.7% Western hunter-gatherer ancestry, 49.8% Eastern hunter-gatherer ancestry, and 6.5% of Caucasian hunter-gatherer ancestry, without any trace of early European farmer ancestry. Her mitochondrial haplogroup, the U4b, is characteristic of the Neolithic Ukraine. Today, the highest frequency of the U4 supraclade is in Russia among the Slavic and Caucasian people, including ethnic groups such as Chuvash, Tatars, and Bashkirs. Way of life. As a member of this riverine community, she likely lived a semi-sedentary life. Her community relied heavily on the rich resources of the Dnieper River, practicing intense fishing and hunting of wild game, such as wild boar, red deer, and oryx. This era also marked the early adoption of domesticated animals like cattle, sheep, and goats. Burials at the Vovnigi cemeteries are highly distinct. Individuals were often interred in collective complexes and heavily covered in red ochre. They were typically buried with status symbolizing grave goods, including bird/animal bone tools, shell beads, pottery shards, and pendants made from deer or boar teeth. The I1732 sample was discovered inside a large multi-person collective burial trench typical of the Dnieper-Mariupol river fork. Individuals in these trenches were usually placed side by side or sequentially over time as the community returned to the site. Like the other members of her group, she was laid out in an extended supine position, flat on her back with legs straight. The Vovnigi cemeteries were thoroughly excavated by Ukrainian archaeologists investigating the rich prehistoric landscape of the Dnieper Rapids before and during regional development project. Her skeleton and the immediate surrounding soil were heavily saturated with red ochre, a natural iron oxide pigment. This mineral was powdered and showered over the deceased during the funeral ritual, leaving a deep reddish stain on the bones and grave floor. The graves at Vovnigi too feature unique pastoral and hunting status items. On or directly surrounding the bodies, archaeologists found pierced shell beads, animal bone tools, and pendant plaques made from the split tusks of wild boars or deer teeth. Clustered around the burial area were shards of pointed base or comb-textured Dnepropetrovsk pottery, which frequently held food or ritual offerings for the afterlife. Faunal remains. Surrounding the human remains were bones from wild game and early domesticates, such as Uryx wild boar and early cattle, remnants of a funerary feast or food provision left with the dead. The scientific record establishes that sample I1732 had a heavily aquatic fish-based pescatarian diet supplemented by wild game. Because paleogenomic studies do not explicitly write out individuals' modern food journals for every sample, researchers reconstructed her exact diet using two precise, factual lines of evidence: stable isotope analysis of the bones from her site and the freshwater reservoir effect, directly impacting her radiocarbon data. The Vovnigi population exhibits exceptionally high nitrogen levels. This provides empirical proof that her primary protein source was riverine, consisting heavily of freshwater fish. Her community supplemented this fish diet by hunting wild animals common to the Dnieper Rabbit. Don't know how to say it. Dnieper Rapids forest steppe, including Unyx, wild cattle, wild boar, and red deer, like we said before. Broad dental assessments conducted on the Dnieper skeletons at the site show a complete absence of dental cavities. This confirms that the diet was almost entirely protein and fat heavy, lacking the processed carbohydrates or cultivated agricultural grains like wheat or millet, consumed by contemporary Neolithic farming societies in southern Europe. So, what would this lady's life have looked like? Based on the verified archaeological and genetic data from the Vovnigi 2 site, here's what a typical day in her life would have looked like: a river-centered world. Her entire existence was tethered to the Dnieper Rapids. She lived in a semi-sedentary settlement located on the high northern terrace of the riverbank. The river was the lifeblood of her community, providing a highly reliable and abundant source of food that allowed her people to live in one place for much longer periods than traditional roaming hunter-gatherers. Her daily routine and diet: the morning catch. A significant portion of her day was spent processing freshwater fish, like massive catfish and sturgeon, caught from the rapids using bone harpoons, fish hooks, and woven river nets. The hunt. She lived alongside skilled hunters who targeted Uryx, wild cattle, wild boar, and red deer in the surrounding forest steppe. She would have been responsible for butchering these animals, smoking the meat, and scraping their hides to make clothing. She did not bake bread or tend to crop fields. Her diet was entirely wild, heavy in protein and fats, which kept her teeth completely free of cavities, but subject to heavy wear from grit. Her tools and craft. She used and likely helped craft Dnepropetrovsk style pottery, heavy, pointed bottom clay vessels decorated with distinct comb-like impressions or stamped patterns, used primarily for storing fish oils and cooking stews. Her community relied on high-quality flint imported from nearby riverbeds to make scrapers and knives, alongside heavily polished bone awls and needles for sewing leather garments. Her family and community. The mother's role. She was a mother raising her son, sample I1378, within a close-knit, multi-generational clan. Her skeletal markers indicate a life of intense physical labour, paddling boats, carrying heavy loads of fish and firewood, and enduring the harsh winters of the Ukraine steppe. A sacred landscape. The Vovnigi cemetery was not just a burial ground, it was a sacred landmark her community returned to for generations. She grew up seeing the collective burial trenches of her ancestors. When she died, her community laid her flat on her back in that very trench, covering her body in brilliant red ochre pigment and leaving behind animal teeth pendants to honour her status as mother and provider within the clan. So I've looked up checking the match between my sample I1732 and sample I1378. The genetic data confirms they are a first-degree mother and son pair, though the researchers themselves note a specific complication with physical evidence. According to the explicit wording from the David Reach Laboratory Supplementary Data 2018. While this article was in press, we identified genetically that I1732 is likely the mother of I1378, although we note that the radiocarbon dates for these samples are inconsistent with this identification. The concrete facts. The DNA match is indisputable. Genome-wide analysis looks at the exact amount of shared DNA blocks, called segments of identity by descent. The genetic sharing between I1732 and I1378 is so high that they can mathematically only be a first-degree parent-child pair. Because I1732 is molecularly confirmed to be female and the other is male, she is genetically his biological mother. The radiocarbon dating paradox. Their physical bones yielded wildly different carbon dates, nearly 800 to 1,100 years apart on paper. A mother cannot be born a millennium before her son. The resolution. Later independent genomic investigations published in BioRxiv 2020 applied temporal population structure tools to bypass the flawed radiocarbon data. These tests proved that their actual lifespans overlapped to within a few decades, confirming that bone contamination or massive local reservoir effects caused by the heavy fish diets had severely corrupted the initial radiocarbon dating of their bones. So mother and son, their two skeletons were not buried side by side or embracing in a single grave. They were uncovered as distinct separate skeletons within the large communal cemetery trench at Vovnigi 2. The relationship was completely invisible to excavators. When Ukrainian archaeologists dug up the site in the mid-20th century, they had no way of knowing these individuals were related. The skeletons were given entirely separate designations based on their positions in the excavation grid. Sample I1732 was designated as burial M32. A purely laboratory discovery, their biological bond was only discovered decades later in 2018 inside a genetics lab by comparing their extracted DNA data on a computer. Because their physical bones were handled, catalogued, and stored as individual museum specimens long before the DNA test, there was never a moment where the two skeletons were laid out together for a photograph as a mother and son. But the DNA proved they were. So again, the tests done on Genome Link are autosomal, so it can be either from my mother or father's side. I don't know my father's DNA group. I only know my mtDNA. So does it match in with my mtDNA? It could be one or the other. So your mitochondrial DNA, haplogroup J1C3, and her haplogroup U4B do not connect directly. Instead, they share a very ancient common grandmother who lived in Western Asia during the Upper Paleolithic period, roughly 45,000 to 50,000 years ago. This shared maternal ancestor is known as in genetics as haplogroup R. From the single prehistoric woman, haplogroup R, the family tree split into major branches, then went on to define entirely different chapters of European history. From haplogroup R came haplogroup JT, came haplogroup J, then J1C3, early Neolithic farmer. From the same haplogroup R, you get haplogroup U, the split off, haplogroup U4, and then her one, U4B, Eastern hunter-gatherer. Your maternal line and hers represents the two distinct competing populations that collided in Stone Age Europe. Her line, the indigenous foragers, her branch U is one of the oldest native hunter-gatherer lineages in Europe. Her specific subclade U4 became highly concentrated among the eastern hunter-gatherers of the Ukraine and the Russian steppes. Her ancestors stayed in the north and east, perfecting a riverine lifestyle based on fishing and hunting wild game. Your line, J1C3, the early farmers, your branch J evolved quite differently. It emerged later in the Near East and the Fertile Crescent. The subclade J1C is deeply associated with the early European farmers. Your maternal ancestors were part of the historic wave of agriculturists who migrated from Anatolia, modern-day Turkey, into Europe, introducing farming, domesticated crops, and permanent village life. The historical meeting point, when sample I1732 was living along the Dnepropetrovsk rapids around 5300 BCE, your farming maternal ancestors, J1C, were rapidly expanding through Central Europe and into Western Ukraine via the linear pottery and early tripillia cultures. While she herself carried pure hunter-gatherer DNA, her community stood right at the geographic border where your agricultural ancestors and her foraging people finally met, traded, and eventually intermarried, creating the mixed populations that formed the later Bronze Age cultures of the steppe. Staying in the Mesolithic too, next on the list to look into is the Neolithic Lithuanian male, Donkalnis 7, the Lake Berzulis, Lithuania. I matched 0.19% to this and genetically closer to this sample than 83.67% of other genome link users.